The overall goal of this method is to describe a three-dimensional alginate-bead culture for maintaining human pituitary adenoma cells.
This method can help answer key questions in the tumor cell biology field, such as the expression of different markers of invasiveness.
The main advantage of this technique is that alginate scaffold, in contrast with other three-dimensional systems, permits pituitary adenoma cells to be liberated from the alginate beads for further investigation.
Demonstrating the procedure will be Carmen Solano-Agama, a research assistant from my laboratory.
To begin, while working inside a laminar flow cabinet, place previously obtained adenoma tumor tissue in a 35 mm dish containing approximately 20 milliliters of calcium and magnesium-free PBS. Using surgical tweezers, transfer the tissue into another petri dish containing fresh PBS to wash the tissue. Repeat the transfer into fresh buffer until the red blood cells and debris are removed. Next, with the surgical tweezers, hold the pituitary adenoma tissue and with a small surgical scissor, cut the tissue into small pieces. Then, using a Pasteur pipette with rounded edges, transfer the tissue fragments and buffer into a 15 milliliter tube. Centrifuge at 68 times G and room temperature for 10 minutes. After the spin, use a Pasteur pipette to remove the PBS and add 3 to 5 milliliters of collagenase solution.